Good morning, everybody, or good afternoon, or good evening, depending on when you're where you're tuning in from. Welcome to the second day of Biotechnology for the Built Environment, or BBE 2021. So yesterday we had presentations based on our microbial environments theme. And today we move on to a second HBBE theme, which is living construction. And the bar was set really high yesterday. I hope many of you were there to pick it up. But if not, I'd encourage you to keep in touch with our YouTube channel and where we'll be uploading the videos from yesterday's presentations. I'm Martyn DadeRobertson. I'm Professor of Emerging Technology at Newcastle University, and I'm one of the co-directors of the Hub for Biotechnology in the Built Environment. And in this context, I'm also the co-lead for the Living Construction theme. I'm Meng
Zhang, and I'm Associate Professor in Microbial Biotechnology based in Northumbria University. With Martyn, I'm co-leading Living Construction theme.
So just before going any further today, just to remind you what we started yesterday, that I mean, you already know that these presentations are being broadcast on YouTube because you're already watching us there, presumably. As I say, they will be recorded and we'll be putting them up later as well as separate presentations. During the course of today's presentation, our guests are going to also have a Q&A session connected to their work. Please use the YouTube chat to put any questions that you've got for them. We have a team of people who are reviewing the YouTube chat and taking out questions and feeding them through to us in Zoom. So we'll be using those as the basis for the conversation with our guests today. Also engage with us through social media using the hashtag BBE21 and through Twitter and Instagram using @biobuildings, which is the HBBE hashtag. And then if you, I mean, you already know you're on our YouTube channel as well, but you can keep in touch by subscribing to our YouTube channel for new videos. Also, the main hub for all of this is our website, www.bbE.ac.uk, where you'll find the timetables for the events today and also lots of other information about things that are happening within the Hub for Biotechnology in the Built Environment. So we have a really exciting day planned, and I'm super excited by the presenters that we have managed to get. We put a call to members of the HBBE a few months ago to ask who their dream team would be to have invited speakers, and we got the dream team here today. So after Meng and I give our presentation introducing the theme and a couple of interesting projects, we've got presentations from Bastian Bayer, then from Jenny Sabin and Peter Nguyen, and we'll be introducing them as the day goes on. I just also wanted to highlight as well in the middle of the day that we've got a poster session, really exciting posters from our PhD students and postdocs. You should by now have received links to their Miro board, so you can go and look at their posters now. And each of the posters has a Zoom room attached to it, so you can click on the Zoom room and speak to the PhD students or the postdocs live there. And I really would encourage people to do that during the lunchtime session today. And then this afternoon, we also have presentations from the 3 research fellows in the Hub for Biotechnology in the Built Environment connected to the Living Construction theme. Just by way of very brief introduction, we wanted to highlight their work. So first up this afternoon, we'll have Jane Scott, one of the research fellows at Newcastle University, who's a specialist in textiles design using a combination of biomimicry, but also biointegrated approaches to fabrication, combining textiles design with biological growth. If you were around for Angela Sherry's project yesterday, you will have seen some of their collaborative work highlighted, and there's some really exciting things going on there.
So we will have Yunhong Jiang, and who is Vice Chancellor's Fellow based in Northumbria University, and his background is about engineering. He has extensive research experience in bio-based construction materials. He also interested about the interaction between microbial cells and nanoparticles, and he has project to apply nanoparticles into novel biomaterials and add functionality in those materials.
And then finally, at the end of the day, we'll have a presentation from Magda Theodorodou. She's a civil engineer working on healing masonry, particularly masonry and heritage buildings using microbial approaches, essentially using microbes to heal existing stone structures. And really exciting work, essentially giving buildings an immune system. And so she'll be talking a little bit about some of her work in this area as well. And she will top out the day. So we just wanted to start by talking a little bit about the composition of the Hub for Biotechnology in the Built Environment. So these are people. So Meng?
Yeah, so in Living Construction theme, so far we already introduced the 3 of our research fellows. And also we have 5 postdoctoral researchers and work on various different funded projects. And also we're going to have 1 more to join us in next couple of weeks. At the moment, we have 12 PhD students directly belong to Living Construction theme. We also have 5-6 PhD students associated with Living Constructions. As Martin mentioned, our postdoc and PhDs have produced posters to present their research project. Do please go to check out their posters during the lunch break.
And I just wanted to highlight as well that, so living construction was one of the original themes that we used to set up the Hub for Biotechnology in the Built Environment. In many ways, it's the theme that's been running for the longest, because we sort of established some of the core principles before the HBBE was founded. But we also, within the HBBE, have a kind of bottom-up approach to devising our themes as well. And there are 2 themes that have come out really as a result of the work of our PhD students that we just wanted to highlight briefly today. So the first of those emerging themes are biofutures for transplanetary habitats. So this really leverages a group of our students who, not through planning on our part actually, but through just general look, are all very interested in space exploration as an application for biotechnology. We work, and in fact, we're now collaboratively working with NASA, but we have a real interest in this area that includes not just technology development, but also overlaps in thinking about humanities and social sciences of space exploration and how biotechnologies might be used, or indeed how they have been used in the past to inform space exploration. So keep an eye on the HBBE website for developments of this theme as we grow it. And the second of these special interest groups has been a kind of long-running project for one of our PhD students, Dylan Oskan, who's been working on mycelium, the root network of fungus, as a way of making materials, but also a way of thinking through biodesign processes. And as a result of that, and a growing mass of people who are really interested in that within the Hub for Biotechnology in the Built Environment, she set up a network called Mycotecture. You can find more about it at mycologyforarchitecture.com. It's a big URL. You can follow her on Twitter. I think mycology4 is the right thing, but Dylan, if you reach out to Dylan, she can let you know. Follow her on Instagram. The first of the events that she's been organizing, she's managed to collect an incredible range of speakers to come to these events completely free, there to build a community really. The first of her events will be taking place on the 29th of July this year, with others planned over the summer and then into the autumn and winter next year. So I just wanted to highlight those. Those are not necessarily only connected, by the way, to the living construction theme, but are interest groups that cross all of these different themes. So back to living construction then, and just to start off by saying what we're about in the living construction theme. And I suppose the easiest summary that I can give is that in construction, we tend to make use of a wide range of biological materials already. We think of timber and limestone and so on, but we utilize them well often after the organism that created them is dead. And our question is, can we fabricate with living systems while the organism is still alive? And indeed, can we make those materials continue to live after they've been created? So it's a complicated thing to do and involves many different facets of understanding. But what we started to do in the work, particularly that Meng and I have been leading, is to develop a strategy which is based on thinking about the way in which biology fabricates materials and splitting those categories into four. So we talk in our group about materials that are constituted of living cells. That means that they're made of living cells. So, for example, mycelium that I just referenced before is an example of a material that is made of the living cells, a root network of the fungus predominantly. Materials that are produced by living cells, so made by a cell, but then exported outside living cells as part of an extracellular matrix. And we're going to highlight the production of bacteria cellulose as part of that process. Materials that are induced by living cells, so materials that are triggered by the release or usually the change and the alteration of the chemical environment of the living cell, such that the material is synthesized, but is separate from the biological process itself. So it's self-assembled outside the cell, independent of the cell's processes, but related to changes that the cell makes to its environment. Or made active by living cells. So that's where we apply living cells to another material and give that material active properties based on the application of the living cell. Now, if you go to the poster sessions at lunchtime, you'll see examples of all of these different fabrication strategies across a range of materials and processes. But we're going to use our presentation now to focus on two of these areas and to highlight the two most important, I guess, funded projects that are part of the living construction theme. Thinking Soils, which is a project that we've had actually predates the Hub for Biotechnology in the Built Environment. In many ways is the model that we used to form the Hub, as in the way that we structure and think about it. We're going to do a bit of a deep dive of that project as well, and presenting some new material that we've never presented before, and giving a sort of overview of some of the quite sophisticated synthetic biology, microbial and microbiology that we're doing, as well as some of our bigger themes and ideas. And then we're going to give a sneak peek to a new project that we have just initiated called Living Manufacture, which is, well, I'll explain, is about developing a new sort of fabrication process based on biology. So we're just going to give those as two examples. There's so many things that we could talk about. We don't have time to do it all, but we wanted to highlight these things. So I'm going to start with Thinking Soils. So Thinking Soils is actually two grants that we've been awarded. The first one, the EPSRC project, Computational Colloids, was a pilot project completed in 2017 that led on to the project that we now call Thinking Soils now, which is due to end in 2022 now. So we've just had an extension on the project. And so the idea is that and I should also mention that it's a collaborative project. So both Meng and I are principal investigators on this project, and we work with Helen Mitriani, who's a civil engineer, and Neil Weipat, who's a professor in the Department of Computing, actually, but a specialist in synthetic biology. And so you'll see some of their work through here as well. So the concept is a straightforward one, but actually very complicated to try and establish. So our idea was that what would happen if you could imagine a self-constructed building foundation. So rather than doing what we currently do, which is when we build a new construction, we go and dig trenches, we fill them with reinforced concrete, and we build on top of those to provide a stable foundation. What if the soil could respond by making materials in response to loading? And so we started thinking, well, can a bacteria sense mechanical change in its environment and then synthesize a material, cause a change in the material in response, such that the soil will strengthen itself automatically in response to load? So that's a simple question, but involves us looking at many different levels of understanding of the microorganisms, how they can be engineered, how they can be used, right from the nanoscale right up to the building scale. So we organized the project, and apologies for this slide, it's not my favorite slide to show, but it is the one that is based on the work package design, we've organized the project as we do now many of our projects around these different scales of the design, starting with the molecular design, the design of the genes themselves that do the pressure responsing, or at least feed in the signal, take the signals from pressure response. Then we translate those into microbial processes that involve mineral deposition to give us our strengthening materials. And then we go to the civil engineering scale to look at how this would affect a large scale building process through both computational modeling and ultimately, we hope, through a physical demonstrator. And we're just going to take you through and give you a little bit of a glimpse into these different processes, starting with the molecular and the nanoscale.
Right, so I'm going to report some progress made from the work packages related to the biological aspect. I will start with Neil's work package. This work is mainly conducted by Polly. Here you can see her picture. And this work package is aimed to design a pressure responsive Bacillus subtilis strain. Next, please. So there are 2 steps to achieve this aim. The first step would be to identify genes which are upregulated in response to elevated pressure. The pressure we chose in this project were 200 kilopascal and 400 kilopascal, which is similar to what we would expect bacteria to experience in soils. The pressurization of the bacteria was performed in the hydrogels, which were made of low melt agarose. The hydrogels have potential to mimic the porous network in the soil environment, and it also being widely used as an alternative media to study soil for various applications such as agriculture and geoengineering. So another benefit of the using hydrogel in sinking soils is that it's easy to collect and visualize the samples, no matter they are the bacteria cells in this work package or calcite crystals in another work package I will talk about later. So the cells were pressurized for 30 minutes to allow us to identify the pressure response without acclimation occurring. Then the mRNA was extracted and sequenced. The differentially expressed genes were calculated and analyzed. So Martin, could you go next? Right, thanks. So this slide shows some result from our RNA-seq, and many differentially expressed genes in 200 kilopascals conditions involved in carbon utilization and secondary metabolites production. This response makes sense to us as 200 kilopascal is only slightly increase in pressure change. So we are seeing a very typical general response to stress happened. However, in 400 kilopascal conditions, we're seeing a much stronger cell stress response and a likely physical strain being put on the bacterial cell wall and cell envelope. So in both conditions, there are 14 differentially expressed genes in common, and those genes show similar patterns, with 200 kilopascal having a consistently higher fold change. Also, among those differentially expression genes, we found there are significant numbers for wear genes. So to the people who don't have a biological background in the audience, I just want to explain what is wear genes. So wear genes are those genes that hasn't been sent a function or have a putative function associated with them. Basically, they were not being studied very well yet. So we're very excited about those wear genes because it's very possible those genes could be very much close or associated with elevated pressure stress. So after looking at those data, we select a few upregulated candidates. Then Polly went back to the genome sequence of the subtilis and tried to identify the native regulatory regions for those genes. This is those regions actually are required for our further research to create a genetic control system. Martin, can we have the next, please? So the second step to create a genetic circuit using identified regulatory regions to give some output only under pressurized conditions. The challenge we faced is that the stress response in the bacterial cells are extremely complex, and often many genes are expressed in response to any stress conditions, not only to pressure stress. This including the genes discovered in our RNA-seq. Some of them could be potentially differentially expressed under anaerobic stress, or salt stress, or other physical chemical stress. So if we only use one regulatory region to create pressure responsive element, the regulated gene is likely to be changed in other conditions, such as anaerobic condition. Then the output will be produced, which is not what we wanted. Therefore, we planned to create a genetic AND gates, which will take into account of two separate regulatory regions as input, and link this element with the urease production. If only one output is present, there won't be any urease be expressed. So this is really important for our system, as we need our system specific to pressure response. Next, please. Now I'm going to introduce molecular biology aspect of the project, which is conducted by Jen. She is engineering a Bacillus subtilis strain with controllable urease expression. Next, please. So urease is a critical enzyme in microbially induced coset precipitation MICP process. It hydrolyzes urea and produces ammonia and bicarbonate. Then ammonia will trigger the increase in pH around the environment, offset the cells, and promote calcium carbonate precipitation in the calcium-rich conditions. So Bacillus subtilis produces urease continuously, and the expression is regulated by nitrogen level. Jen actually show when the media have a very limited nitrogen source, the urease activity actually increased. You can see her figure at the bottom left. However, even at a very increased level, urease activity in Bacillus subtilis is still very low and nowhere near to the urease from bacteria called Sporosacina pastoria, which is the bacteria have been extensively explored in MICP process. In addition, the mechanism for urease activation in subtilis is not well known. As the image shown on the top right here, active urease actually contains 3 structure proteins, also contain nickel ions as cofactors. The addition of the nickel ions require accessory proteins as shown on this figure. And however, if you look at the image at the bottom, look at subtilis urease operon, it lacks gene sequence for accessory proteins. So Jen actually spent 3 years to study the mechanism. However, she didn't have much luck. But if you're interested about this project, please check out her post during the lunch break. Next, please. So to move the project forward, we decided to clone Pastorian urease into subtilis. Eventually, we would like to link this operon with the AND gate produced by poly. And so when the pressure is applied to subtilis, the cells can produce significant amount of urease and then induce the coset precipitation. Next, please. So Jen started with cloning the urease cluster using PHT01 plasmid, and the expression of the recombinant protein can be induced by APTG. If the cloning is successful, we should be able to see more protein expressed at the size range down the bottom left. And as you can see, the SDS page gel on the top right corner on this slide, clearly there's more recombinant protein were produced by 1 millimolar and 2 millimolar APTG. As labeled on the gel, clearly there are more proteins produced in those ranges compared to other control cells. Then she moved on to confirm this result to use a simple urea broth test. In this test, if there are urease present, ammonia will be produced and lead to higher pH. The color of the solution will turn pink. So if you can see the bottom image shows we have engineered cells produce a pink solution compared to the negative control, and also is still relatively faint compared to the Pastorian cells, which is a positive control. Next page, please. Then Jane set up biological replicas to quantify this reaction. Interestingly, although from previous SDS page, we can't really tell the different level of expression by 1 millimolar APTG and 2 millimolar APTG, but clearly there are more ammonia produced by the cells in 2 millimolar APTG settings. If you look at those images on the left side of this slide. And more encouragingly, that we found when we leave the reaction for 72 hours, we can observe amount of ammonia produced in engineered strains start to become comparable to Pastorian urease. So that was really good for us. However, when we conduct more investigation in expression, we found the recombinant urease is not very stable. If you look at the bottom gel image, you will see the expression of those urease start to disappear when we harvest the cells after 6 hours induction. So we decided to integrate those gene clusters into subtilis genome to produce a stable system. And Jane is in the process of doing those experiments. Next, please. So now I'm going to move on to some microbiology and biochemistry aspect of the project to investigate the MICP process for soil improvement. This work mainly been done by Jamie. Next, please. So in this work package, we investigated many parameters which could have an impact on calcite precipitation and consequently the binding properties and the cementation materials properties. This including cell growth, enzyme activity, cell distribution, and different reactance conditions, and also addition of the actives. Although there are many parameters already being studied by other groups for soil improvement, particularly used for the validation of computational models, we found that the existing studies tend to simplify the whole process, and they treated each step as a separate reaction without consideration of the link between different steps. And also there were lots of assumptions being made for those studies. So today I'm going to just present some highlight for Jamie's work. She did some investigation in the liquid solution, and he tested the impact of various chemicals on urease activity. Clearly, the top red figure show the presence of calcium ions have no impact on the urea hydrolysis rate. Then he investigated the rate of urea hydrolysis and the calcium carbonate precipitation. And our figures show that when we have a fixed concentration of calcium ions in the system, the urea hydrolysis rate does not have a linear relationship with the rate of calcium carbonate precipitation. Although this scenario has been commonly assumed as the condition for many computational model invalidation studies. Next, please. Jamie also tried to investigate the urease activity in sand column. If the image on the left shows the lab settings and the scheme for sand column experiments. However, it turned out that it's very difficult. It's very difficult to measure the urea hydrolysis and amount of ammonia being produced in the sand column, as we can show the top right figure. But Jamie found that when the calcium ion and urea has 1-to-1 ratio in the reactant solutions, the utilization of the calcium ions were the most effective condition. This finding is in line with other literatures, and when the calcium ion increased to higher ratio, you can see the calcium ions were not fully precipitated and were also can be detected in the effluent, as shown on the left-hand figure on here. Another interesting observation is when we increase the concentration of calcium chloride in the system, and it leads to the decrease of pH in the sand column. Particularly when we add 400 millimolar into the system, it decreases to pH 7.2, which is really not a favorable condition for calcite precipitation. Can we have the next slide, please? So Jimmy also conducted some investigation on MICP in hydrogels. He investigated the effects of changing reactants composition on calcium carbonates, quantity and morphology. As you can see, the figures and the table show on these slides, there's many conditions he studied, and there lots of information need to be unpacked from this figure. But the key findings from this set of experiments is not all the optimized conditions we investigated in the sand column are applicable for the hydrogels. The difference observed between these two media could be due to many factors, but we believe the different porosity and permeability play important role in those differences. But those different porosity and permeability could be the case when the MICP are applied into different soil types. So we think this should really be taken into consideration by geoengineers and computational modelers when MICP are studied for soil improvement in the future. So also in the hydrogel, we can observe the increase of the production of calcium carbonates along days passing by. And on the right-hand images, you can see the crystal formation at different days. And also, when you zoom in, you can see many cells be buried in the crystals. Jimmy's still in the process of finish off those experiments. And the idea is those data from those experiments can be fed into Genius computational model. Now I'm going to hand over back to Martyn to talk about other aspects of the project.
Thanks, Meng. So you can see that we've got, Meng, I'm getting feedback from your mic, so you might want to mute yourself on there. So you can see that there is a lot of complex layers of biology going on, from the molecular biology through the microbiology to the in situ biology, as it were. So the biology that is being created within our test soil conditions. And so we've got to try and pull that all together. And this is really the mission of the final year of the project. And so we're going to bring it together in two ways. So one is through an integrated computational model. So actually measuring forces through soils is something that that is done routinely in civil engineering, in geotechnical engineering, but the kind of model that we need for this kind of analysis is more complex than the ones that we typically use in civil engineering. So when a soil is loaded, of course, the load is distributed through the soil volume, but what we're actually interested in is the increase in water pressure within the saturated soil. So we expect that our bacteria are actually going to be responding to that increase in water pressure rather than necessarily the act of directly loading onto their cell. So without going into a lot of geotechnical detail, what happens when you load a saturated soil is you get a period of time where the water pressure will go up, then the water will dissipate through, and so you'll get that water pressure dropping again, and eventually the load is taken by the skeleton of the soil, and that changes depending upon the characteristics of the soil that you're working with. So the first thing is to start to develop a model which can do that analysis of the loading of the soil, and then translate that into something that we can integrate our biological data with. And this is what Jingye Wang, our research associate, has been working on over the last year, building on models that we previously produced in this project. And so I'm not going to go into the detail of every layer of this model, but to say it's a complex and multi-layered model. It starts with the loading conditions given a set of soils, given the amount of saturation within the soil, and known values for the stiffness of the soil and so on, how it responds to those loads. And then we add in our layers of biological information, and that includes things like cell distribution, so how many cells we'll expect to see in different layers of the soil, and then this genetic response mechanism. So it's not just like a switch where it's on or off. Actually, there's a degree of responding to the particular conditions that is variable depending on the condition that you find the bacteria in. The production then of the urease, and then that translation of that urease ultimately into calcium carbonate that then binds the soil together. Then importantly, that's a dynamic model. So as you're loading and as the bacteria are responding, that's also then changing the properties of the soil itself. So there's a feedback relationship that goes from the initial quality of the soil through these biological processes, and then back to in effect the condition of the soil again. So that's quite a complex and difficult model to build, but we're just getting to the end of the point now where we've got the model such that we can put in our experimental variables. Our overall aim is to then be able to visualize the results. And so this is from an earlier prototype that I developed that maps on different genetic responses from the bacteria to different cementation profiles. So in this case, these sort of the things that look a little bit like ice cubes here are representing different levels of gene expression, and there's then connected to different levels of calcium carbonate precipitation within a volume. This is a volume of 10 meter by 10 meters. And so we not only want to use this as a sort of modeling tool, but also as almost like a computer-aided design tool. So we can actually design bacterial responses that are specific to the loading condition that we're placing on it. So that's the computational model. And the final part of this is then to also build a physical demonstrator. And so we don't have the physical demonstrator yet, but we're getting close now. So we've got our pressure sensor, or we think we've got something that we can characterize as a pressure sensor. We've got our controllable urease expression system, and we understand how the bacteria will work within a soil matrix. We want to bring those systems together into one single demonstrator where we can load in this case, our hydrogels, and those hydrogels will respond by making calcium carbonate crystals where those loads are applied. And so for that, we're introducing our new postdoctoral researcher, Akram, who will join us on Monday. I suspect she is listening to this now, so she now knows what she let herself in for, whose job will be to start to pull all this stuff together into a single demonstrator. And we hope that we'll be able to complete that in the next year. If we do, then it will be a really groundbreaking project and something that represents a very a kind of new area of actually material science, as well as a new area of construction technology as well. I also wanted to recognize that as well as the core project, we have some spin-offs, if you like, from Thinking Soils. So we've actually, I think, supported 4 PhD students at this point, one of whom is completed, a second of whom has submitted their PhD, and Thora is coming up now to submitting her PhD pretty soon as well. And she's taken working with the sort of things that you saw Jamie working on, which is this characterization of bacteria within soil-like volumes, in this case within sands, to understand how the bacteria behave, how the calcium carbonate crystals alter the characteristics of the materials, and started to use that as a way of developing a novel casting system using microbes to induce calcium carbonate formation by seeding the bacteria within sand volumes and then pumping reactants and food through the system until you get these cemented volumes, these blocks that you see. Experimenting with different ways of doing that, not necessarily to make homogeneous blocks, but to think about the dynamics of casting potential. So using a design approach to think about the way in which we might cast more complex shapes and forms, possibly with functional gradation and different properties. She's also been developing novel ways of visualizing those results using a combination of 3D modeling, 3D scanning to understand the dynamics of that process over time. So how do you build up different calcium carbonate precipitation in particular places in relation to where the bacteria are, the flows of reactant, the flows of nutrient through those volumes. And we're actually using this as the basis for a new grant that we were in a meeting this morning, putting the final touches to a grant that we're submitting in a week or two's time that develops this project much further into what we think it may be an industrially scalable manufacturing system for biomineralized materials. So just to finish off that, I want to acknowledge, we've acknowledged Helen and Neil already in the project, but we also have people who worked with us in the past, as well as the work package postdoctoral and PhD students that you've seen. Beate, who had previously been doing the work that Jamie is now doing. Javier, who was our first PhD student to come through the Thinking Source project. And also we're very grateful for the advice of Henk Jonkers at TU Delft, who's an advisor for the project. And just to reiterate that we're funded from the Engineering and Physical Sciences Research Council, the EPSRC. So the second project we just wanted to share with you, and this one we're going to share much more briefly, is a project we call Living Manufacturing. In fact, its specific name is Principles for a Microbial 3D Printer. I'm getting that right, Meng. Yeah, I'm getting that right. This is a much earlier stage project, so we're only a few months into this one. But this is in our materials that are not induced by bacterial cells, which is what Thinking Cells was, but are made by are produced by living cells. So Meng and I both lead on this project with a team that includes Thora and Josh, who you'll meet later, and both of whom has posters in the session today as well. So the animation that's running here is a concept work that we did as part of the grant application. So what you're seeing is a bioreactor connected to a computer and a projector. So the bioreactor is a big box, really, that we fill with a liquid. So what we're trying to do here is to fill the bioreactor with a liquid that contains a bacteria capable of producing cellulose. And is fed through this process as well, so it's given nutrient. We've got a computer loaded with a 3D model, and then the 3D model is sliced and translated into a projector. So the idea here is that our bath of bacteria, the green ones are the cellulose producers. So if you've ever worked with microbial cellulose before, you'll know that the bacteria produce these cellulose polymers. They excrete outside the cell, and then you get on the top of the liquid culture a pellicle forming, not much like this, actually. This is a slightly hokey graphical representation of how it works, but you effectively get a skin forming. And then within our bath, we're going to have another type of bacteria that is sensitive to light and produces a pigment in response to light which is shined on it. So the light being shined on that surface is coming from the projector, and the idea is that that light changes depending on the height of the pellicle. So we want to go really thick pellicles, and as we do so, we're triggering this polymer production in 3 dimensions, such that at the end we have this thick, probably not that thick. We expect to be able to grow pellicles of maybe 2 or 3 centimeters, but that what will happen as the pellicle is dried out, you'll get this cellulosic material, which when you open up will reveal the 3D the imprint or the shadow, if you like, of the 3D object through this microbial precipitation process. So that's the concept behind the project, and so we've received a pilot funding to do it, and this is really part of a much bigger process that we think might be the basis of a new type of fabrication system. So while we call this a 3D printer, it actually has a lot more to do with biological growths, and this is more like a bioreactor than it is a 3D printer, because the cellulose is self-assembled within the 3D printer. So we're not trying to extrude a material, we're growing the material within the bioreactor. And our proposal is that much like you would with a 3D printer, where you might have cartridges of material, actually what in this case you would have are cartridges of bacteria, each that are engineered to respond to their environment a particular way. So that might be a light shining at them, as in the image, the example I showed before. It may be a chemical trigger that we can then interact with and control as the pellicle forms, such that you can begin to develop quite complex and multifunctional, functionally graded materials. So you can imagine that within the cellulose, you may make mineral crystals forming, or you may get other sorts of polymers, or indeed you might break down the polymers as they form to create selective weaknesses within the material. And we think that that could be the basis for all kinds of interesting applications, not only for architecture, but possibly for fashion design and for medical devices and other things. So this is really the first step towards establishing the principles for this system. And we hope that we'll get some really interesting results over the next couple of years. So to kind of conclude that part of the work, then the idea of these different approaches is not to see them in singular terms, but to integrate them together. So we think that the future of this way of fabricating and constructing with biology will require us to think about all of these different fabrication strategies, not independently, but together in single systems. And the way we've been increasingly conceptualizing this is through the development of systems that integrate, well, I think you've seen examples of that today, that integrate what we're describing as wetware, the biology, sometimes engineering the microbes to work in, for example, in the work that we've shown you, engineering the microbes to work in ways that we want them to, but integrating those with hardware, so the development of novel bioreactors or fabrication type technologies. And we have examples of that both in Thinking Soils and the Living Manufacture project as well. And also software used to model, but also used to define the parameters for our fabrication process, creating an integrated approach. And we see this as being very much the future direction that we're wanting to take. So just to conclude with a shameless plug, if you're interested in some of the theories and the underpinning ideas, I have a book, Living Construction, which came out at the end of last year. It's a slim volume. It's actually going to be part of a series. So we have 3 or 4 books that will become part of the series, a kind of collector's set, if you like, that are going to come out over the next couple of years from various authors. Mine is the first, and I'm editing the series as well. They are slim volumes, but packed full of good stuff. I would also say that if people are interested in contributing, the books are about 30,000 words, and they're really focused on a broad range of biodesign, so not just living construction type projects or biotechnology for the built environment, but biodesign as an emerging field. If you have a book idea, perhaps you're a PhD student coming to the end of your work and think that you've got enough for a distinctive volume, then do reach out to me and I can discuss with you how to apply for that. And yeah, and that book is available from Amazon and other retailers, as well as an ebook or indeed a free PDF if you email me and ask nicely. So that brings us to the end of the session. We have a little bit of time, and I noticed that there have been some questions coming through the chat, Meng. So before we go on to introduce Bastian, do you want to, if I field the questions? And I think the questions have predominantly been biological. So it may be that you have to answer, and I should say that we can't necessarily answer them all because some of these are other people's work packages, so we can't necessarily know, and some of it is we're still waiting to publish, so we'll go with that. But if we start, we've got a question here, is asking us if we can give any more detail in the increased stress responses at 400 kPa. So a question of, did the bacteria sporulate under pressure in the Thinking Soils project?
The answer for that question is no, actually, and we think why it's not, because I think 400 kilopascal is still relatively low increase the pressure, so they haven't triggered the sporulation for the cells. Probably along to the second question.
Yeah, I just to say there that one of the things that was interesting is that when we started the Thinking Soils project, we assumed that people had already done pressure studies for bacteria, and of course they have done pressure studies for bacteria at very, very high pressures or very, very low pressures. So there's been a lot done at sort of, can a bacteria survive a vacuum by sporulating and surviving in that way. There's been a lot done connected with pressure pasteurization. So when you get this idea that you're using pressure as a way of pasteurizing, we know, for example, E. coli survives that kind of pressure very, very well and has excellent stress responses. We're talking about relatively small, elevated pressures for the bacteria, and it's a question of whether they do have any sort of genetic response at all. We couldn't find anything on that, and the research has actually shown that yes, they do, and they're quite distinctive to these different pressures. So yes, question 2. Do you have that? So the question is, from
the secondary metabolites, we didn't see much antibiotic associated production, but we definitely saw a loads of citrifers related protein genes exchange, and actually there's a group of genes involved in iron starvation and biofilm formation. Yes, the answer is yes. There's definitely see the citrifers production change in the secondary metabolites production.
And the third question is, are we thinking about looking into other microorganisms in the future? Yes, absolutely we are. So there is some of this which is now part of a couple of grant applications that we're working on at the moment. There are a number of really interesting organisms. We've gone with Bacillus subtilis and tried to engineer it because it's, for those of you who know about synthetic biology, it's a good chassis organism. It's known as being an organism that we can work with. And we've also worked on this for our pressure sensing. It's just a lot easier because it's genetically tractable, well known, and we've got lots of tools that allow us to use it, and our collaborators are there. However, there are lots of really interesting, and not only calcium carbonate formations, but other sorts of mineral crystal formers as well. And we're kind of actively looking at ways in which we can utilize some of these organisms in the wild and also engineer them to work in different ways as well. Don't know how much we can say about that, Meng, but that's
just the add-on. We got a great asset in HBBE. We got Paul James. Probably you will hear his presentation tomorrow. And he is a synthetic biologist working with non-model organisms. So that significantly extended our possibility to work with non-model organisms like Bacillus subtilis. So as I mentioned, as Pastorium are the bacteria extensively explored in MICP process. And actually we are actively looking at genetically modified Pastorium. They add different function into that bacteria cells, and hopefully they will have all kinds of different functions. We can add it into the MICP process.
Great. So that's the end of the questions, and so thanks for that. So this went a lot quicker than it did in rehearsals, Meng. So we are now ahead of time, which is not what I was expecting to do. We could have kept in the extra slides. But thanks very much for the questions. If you've got any further questions you want to fire through, use the YouTube chat. We'll try maybe to answer them via text if there are particular ideas. But if you're really interested in this project or the spin-outs from it, we do encourage you to contact us. We're involved in a lot of different groups who, for example, using some of our bacteria species and so on in their own work. So we're very keen to hear about people who are interested.